Good day, this is the Professor, CBMD PhD, and today's Monday, February 13th, 2023. It is 11:23 and 24 seconds, p.m. Eastern Standard Time, and today's research, genetic disease. So let's jump to it here. As protein droplets may cause many types of genetic disease, date February 8th, 2023, source, Max Planck, Gazelle Shaft. Malfunction of cellular condensates is a disease mechanism relevant for congenital malformations, common diseases, and cancer, new research suggests. As patients with PPTA syndrome have characteristically malformed limbs featuring short fingers and additional toes, missing tibia bones in their legs, and reduced brain size. As researchers found out, PPTAS is caused by a special genetic change that causes an essential protein to migrate to the nucleus, a large proteinaceous droplet in the cell nucleus. As a result, the function of the nuclear condensate is inhibited and developmental disease develops, the researchers say. What researchers discovered in this one disease might apply to many more disorders. It is likely not a rare unicorn that exists only once. They just could not see the phenomenon until now, because they did not know how to look for it. And contributors from all around the world, the team is pushing open the door to new diagnoses that could lead to the elucidation of numerous other diseases, as well as possible future therapies. They discovered a new mechanism that could be at play in a wide range of diseases, including hereditary diseases and cancer. In fact, they have discovered over 500 similar mutations, 101 of which are known to be associated with different disorders. The actual work is just starting now. They will find many more genes with such disease-causing mutations that can now test their mode of action. Affected individuals have a complex and striking malformations of the limb space and surface and bone systems, only partially described by the already long disease name brachycephaly, polydactyly, tibial aplasia, hypoplasia syndrome. With fewer than 10 documented cases worldwide, the disease is not only rare, but ultra rare. As to track down the cause, the researchers decoded the genome of 5 affected individuals and found that the gene for the protein HMGB1 was altered in all patients. And this protein has a task of organizing the genetic material in the cell nucleus that facilitates the interaction of other molecules with the DNA, for example, to read genes. In mice, a complete loss of the gene on both chromosomes is catastrophic and leads to the death of the embryo. In some patients, with only one copy mutated. However, the cells are using the intact copy on the other chromosome, resulting only in mild neurodevelopmental delay. But the newly discovered cases did not fit this scheme. All 5 unrelated individuals featured the same ultra-rare disorder and had virtually the same mutation. This is why they are sure that the HMBG1 mutation is the cause of the disease. However, at that point we had no clue how the gene-produced functionality caused disease, especially given that lots of function mutations were reported to result in other phenotypes. A closer look revealed that different mutations of HMBG1 have different consequences. The sequencing data show that in the affected individuals with the severe malformations, the reading frame for the final third of the HMBG1 gene is shifted. After translation of protein, the corresponding region is now no longer equipped with negative, but with positively charged amino acid building blocks. And this can happen if a number of genetic letters, not divisible by 3, is missing in the sequence, because exactly 3 consecutive letters always code for one building block of the protein. However, the tail part of the protein does not have a defined structure. Instead, the section hangs out of the molecule like a loose rubber band. The purposes of such protein tails, also called intrinsically disordered regions, are difficult to study because they often become effective only in conjunction with other molecules. So how might their mutation lead to the observed disease? And these droplet-like structures behave much like the oil and vinegar droplets in a salad dressing, composed of a large number of different molecules. They are separated from the surroundings and can undergo dynamic changes. They think condensates are formed in a cell for practical reasons. Molecules for a specific task are grouped together in this way. Say to read a gene. For this task alone, several hundred proteins need to somewhat make their way to the right place. Intrinsically distorted regions, which tend not to have an obvious biochemical role, are thought to be responsible for forming condensates. Giving an example to describe how important the physical properties of the protein extensions are in this regard. They can easily make a ball from many loose rubber bands that holds together relatively tightly, and that can be taken apart with little effort. A ball of smooth fishing line or sticky tape, on the other hand, would behave quite differently. As the nucleolus within the cell nucleus is also a condensate, which appears as a diffuse dark speck under the microscope. This is where many proteins, with positively charged tails, like to linger. Many of these provide the machinery required for protein synthesis, making this condensate essential for cellular functions. The mutant protein, HMGB1, with its positively charged molecule, tail is attracted to the nucleolus, as well as a team observed from experiments with isolated protein and with cell cultures. And since the mutated protein region has also gained an oily, sticky part, it tends to clump. The nucleolus loses its fluid-like properties and increasingly solidifies, which researchers were able to observe under the microscope. And this impaired the vital functions of the cells with the mutated protein. More cells in the culture died compared to a culture of cells without the mutation. The research team then searched databases of genomic data from thousands of individuals looking for similar incidents. In fact, the scientists were able to identify more than 600 similar mutations in 66 proteins, in which the reading frame had been shifted by a mutation in the protein tail, making it both positively charged and more greasy. Of the mutations, 101 had previously been linked to several different disorders. As for a cell culture assay, the team selected 13 mutant genes. In 12 out of 13 cases, the mutant proteins had a preference to localize into the nucleolus. About half of the tested proteins impaired the function of the nucleolus, resembling the disease mechanisms: brachycephaly polydactyly, tibialoplasia, hypoplasia syndrome. However, congenital genetic diseases such as brachycephaly polydactyly, tibialoplasia, hypoplasia syndrome are almost impossible to cure, even with our new knowledge. And because the malformations already develop in the womb, they would have to be treated with drugs before they develop. This would be very difficult to do, as tumor diseases are also predominantly genetically determined. Cellular condensates and the associated phase separation are also a fundamental mechanism of the cell that also plays a role in cancer. The chances of developing targeted therapies for this are much better. And this is a professor of microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.